Hello everyone, this is Karamveer, a research scholar from Department of Bioinformatics, Pondicherry University. So today I'm going to present on oncogenic and non-oncogenic class prediction for human papillomavirus using genomic data and machine learning approaches. So human papillomavirus is a double-stranded DNA virus which belongs to Papillomaviridae family, and it used to cause warts and carcinomas. Till date, more than 220 types of human papillomavirus has been discovered, and some of them are asymptomatic and some of them used to cause carcinomas. Based on their genotype, they can be lethal as they can cause penile cancer, head and neck cancer, and other enogenetical cancer. And some of them are asymptomatic. However, HPV and cervical cancer relationship is well studied and it is well established. With advancement in technology, new HPV types are adding every year to the database, and as the computational power is also increasing, such as the methods such as machine learning models are being used in biological sciences. It is important to analyze these data to find a relevant pattern in the data using machine learning models. So in past few years, genomic data and codon users information is being used with machine learning models to annotate the genomes. So here what we have done is we have used RSCU information to categorize these two classes, to classify these two classes. So you can see this is non-carcinogenic and this is carcinogenic class. So here we have used 6 core genes from HPV genome, which are present across the mammalian genome. So E1 and E2, which are involved in replication and transcription, and these E6 and E7 are present in, they are oncoprotein, and 2 of them, L1 and L2, are capsid proteins. So what we have done is we have taken 6 of these proteins. We have constructed an RSU table, based relative codon, synonymous codon usage table, which will tell you the codon biasness. So based on the RSU value more than 1.6, we have constructed a table, and from there we have summed up based on the wobble nucleotide at the 3rd position. So if A on 3rd position, if adenine is there, so all the codons which are having adenine, we have summed up the RSC value. So this is how we have constructed for 4 wobble nucleotides for each gene. So we got 24 features in a feature table for 6 of the genes, and we have fed this to machine learning model, which was able to classify the carcinogenic and non-carcinogenic class of the HPV. For this, we have used linear and non-linear model combination, such as linear regression and linear discriminant analysis, KNN, and naive Bayes and SVM. So we have found out, based on training and testing data, we have found out SVM and logistic regression performed well, and we have hypertuned the SVM model, and we find out that linear kernel with scaled gamma performed based on our test data set. Logistic regression also performed almost same, as you can see. At the end, we find out with SVM, the model accuracy on 5-fold cross validation, it was 98%, 98.4% on training data set and 98% on test data set. The MCC value on test data set was 90.91, which is 91%. The specificity and sensitivity, which are the important measures in machine learning model accuracy evaluation. So we are having specificity 100% and sensitivity 98%. And balanced accuracy on our imbalanced test data set was 99%. And as you can see in our confusion matrix, we are having 6 of the carcinogenic sample classified correctly, out of 57, 56 non-carcinogenic classified correctly. Only one non-carcinogenic is classified as carcinogenic. So in our initial data set, we were having 18 carcinogenic and 129 non-carcinogenic. At the end, we created a balanced data set by oversampling the minor class, which is carcinogenic. So 129, a balanced data set we have created and we tested on our test data set. So what we concluded out of that is linear models such as SVM with linear kernel can be used, such as logistic regression. Linear models can be also used for HPV class prediction, and genomic composition information such as codon usage information can be integrated into these models to classify newly discovered HPV phenotype. And these models can be helpful for the clinician in early diagnosis of novel HPV. So we have got the ROC value on test data set is almost 100%. So we would like to thank DBT for providing financial assistance in the form of DBT Wing Fellowship. Thank you, everyone.